work in our laboratory is also focused on understanding the immune cells within the bone marrow microenvironment of myeloma. As we are all aware, as is truly shown in this meeting, that the tumor cell in itself is not the only culprit. We have the bone marrow accessory cells that really help tumors survive. And one of the examples is dendritic cells, in particular plasmacytoid dendritic cells. We shown in our study in 2009 and also a number of other subsequent studies that the plasmacytoid dendritic cells are the immune cells, and they are innate component. They help generate the immune activity against the tumor cells. But in myeloma, we found that these cells have lost their immune function. They are no more generating any anti-myeloma immunity. Instead, what they're doing is that they are triggering the growth of tumor cells, helping them survive and confer drug resistance. So this is altogether a new finding. A one cell subtype, plasmacytoid, or we call them pDCs, are now able to become an ally of tumor cells and help it grow. So we started focusing on strategies how to target pDCs and pDC myeloma interactions. And in that effort, we started working on one of the drugs called SL401, which at the end is able to really kill pDCs, which are abnormal in myeloma. Suffice to say that these pDCs are much higher in number in myeloma patients than in normal donors. So we have a strategy using this drug to really kill these abnormal pDCs transiently. But we have observed there is an ongoing clinical trial of this agent at Dana-Farber and other multi-center centers, and it's already showing safety to an extent and a response, so which will be presented in upcoming ASH meeting. But the moot point here is that we can really deplete pDCs from the marrow and thereby allow the tumor cells to have effect of the drug on the tumor cells. So basically, you cut off the survival mechanisms through the pDCs by depleting pDCs in the bone marrow. So we have gone on to also understand, can we really generate an immune response, and can we revive or revoke or restore pDC's immune function? And we have started using strategies. For example, we have used toll-like receptor agonist 9 and 7, and we have shown that once you activate these pDCs, they are matured, and after the maturation, they are awakened, they regain their ability to trigger T cell activation and proliferation, as well as then induce cytotoxic T cells, T lymphocytes against the myeloma cells. This is beautiful. The reason is now you have an immune awakening in one part. You are stimulating the pDCs, and thereby you are stimulating the immune response. And that we have also gone on to see whether we can check the checkpoints on the pDCs and the tumor and block that inhibitory signal also. So suffice to say, the therapies that we can envision is to combine agents that can stimulate the activation of pDCs, just like toll-like receptor 7, 9, as well as combine it with the agents that are checkpoint inhibitor like PD-L1 or PD-1 that mediate the myeloma cell and pDC interactions. So many of these checkpoints have been shown to really also affect the T cell and pDC's interaction, as well as NK cells. Overall, in our studies, what we have shown that in this model, you can really induce pDC maturation and activation and have CTLs generated against the tumor, as well as NK cells mediated cytotoxicity against the tumor. So this is the functionality that we have shown, a possibility that you can really invoke an immune response in these patients together with, in combination with other therapies. This is what is the need of the day. And we all know that the CAR T cells and other immune approaches or antibodies are working great, and none of them is as final as or as to the standard of care right now as we would want to. But nonetheless, the research on our side is focused on bringing out new concepts, new understanding of the microenvironment, and new cell types that could eventually be helpful in restoring all the anti-myeloma immunity, and importantly, understanding the myeloma biology. I mean, until 2009, until we published the study, the pDCs were not known to be playing a role. So we had to really dig deep into the myeloma biology to understand what these cell types are. So we discovered that this is a new cell type that is affecting myeloma progression. So I think we have several advances in the basic science in terms of myeloma biology that could lead to understanding of mechanisms and as well as identifying novel therapeutic targets for future clinical trials.